All right, so the last video for the carbohydrates chapter is going to focus on disaccharides and polysaccharides. Really, this is going to be a rapid overview. If you go on and take biochemistry, you're going to cover this in more detail. So instead, what I'm going to do today is just go over the key features of these that you should be aware of as it relates to organic chemistry. So let's take a look at this first disaccharide. This is maltose. It's a disaccharide because it's composed of two different sugars. And if we look at it, you can see that right here in between, we've linked them in a 1,4 manner, meaning the anomeric carbon in this first carbohydrate is carbon 1, is reacting with carbon 4 on the second carbohydrate. And this bond right here is called a glycosidic linkage. So this is called a glycosidic bond. And that's the attachment point between these two carbohydrates. And in fact, we saw how to make O-glycosides in our previous video. So the alcohol that was formerly coming off this position just reacted with that anomeric site, and we were able to link these two together. Normally this happens through a biological process though. Additionally too, if you notice, this bond right here is pointed down. So what we like to call this is an alpha linkage. The way I always remember that is alpha is axial, right? So in this case, it's that axial point that's attaching through the glycosidic bond. Therefore, it's alpha linked, and you can see that this is an alpha glycoside, which is how we describe these. You can have other variations of this that aren't 1,4, but 1,4 is pretty common for situations like maltose, right? The good thing is that maltose is found in a lot of food products. And these alpha linkages are fairly easily broken by our body. So let's make a note. Alpha linkages are easily digested. So we have enzymes that can clip these up, and then we can metabolize those monosaccharides once that bond is broken. All right, let's go to the next one. The next one's pretty interesting. It's lactose, right? So again, this is going to be a 1,4 linkage. This is the anomeric carbon 1. This is carbon 4 and the other one. We've got this glycosidic bond right here. But if you notice, instead of it being axial, this bond coming off that anomeric carbon is equatorial. So this is a beta linked glycoside. And because of that, these are less easily digested. So most human beings do have an enzyme that can break these bonds. It's called lactase. And when this bond is broken, we can metabolize it no problem. However, there may even be some of you in the class that are lactose intolerant, meaning you can't handle dairy products that contain lactose sugars, right? The reason behind that is your body is just not making enough of that lactase enzyme to metabolize all of this. So this sugar goes uncut all the way into your intestines. And when that sugar lands in your intestines, there's a bunch of bacteria in your intestines that starts breaking down this lactose. And when those bacteria break down lactose, it tends to produce a lot of gas, which is why you might not feel super great if you're lactose intolerant and binge on ice cream or milk. So because this has that beta linkage, it's less easily digested than a disaccharide like maltose. All right, so pretty interesting. Let's take a look at the most common sugar though. In this case, what we've got are two different ring sizes, right? So over here, we've got the furanose ring, and over here, we've got the pyranose ring. And if we look at this, this is over here, glucose, and over here is fructose. So sucrose is your standard table sugar that you have in your kitchen, composed of a glucose monomer that's glycosidically bonded to a fructose monomer. And if we look at the anomeric carbon, we can tell that the anomeric carbon would be right here. So let's identify the linkage, right? We know that this is the attachment point. But off that anomeric carbon, we can tell that this is axial. So this would be an alpha linkage. And like I said, our bodies are very well equipped to tackle these alpha linkages, so we have no problem digesting table sugar, whereas some of us have problems digesting milk sugar like lactose. All right, so these are the most common disaccharides. They're the ones you'll see a fair amount if you go on and take biochemistry. But if you go on and take other courses like botany, you'll see different types of sugars, primarily this sugar. So this is cellulose. You've probably talked about it a lot in biology. But really all cellulose is it's a bunch of repeating glucose monomers that are all linked up together, right? And if we look at it, we've got these glycosidic linkages between each of these glucose monomers. So the anomeric carbon here, here, and here forms the glycosidic bond with the hydroxy group coming off of carbon 4 of an adjacent cellulose or an adjacent glucose monomer. Additionally, if we look at this, you can see that this is now beta linked. Because it's beta linked, we can't really digest this. In fact, most mammals struggle to digest cellulose. But if you've studied this in your biology class, you probably learned about how cows and other mammals can digest cellulose, but they do that with the aid of gut bacteria. So it's the bacteria in our intestines or stomach that helps us to digest some of these. Humans don't really do a good job at this, so other animals have evolved to take advantage of this. But kind of interesting to see that it's a subtle difference in this glycosidic linkage, just axial versus equatorial, that makes a world of difference here. If we go down and we look at this next one, this is amylose. Again, this is a carbohydrate that's made up of glucose monomers. So it looks a heck of a lot like cellulose. But the only difference in this case is this glycosidic bond between the glucose monomers now is now all axial. So this is an alpha linkage. And plants make these too. This is also known as starch. So if you eat something like a potato has a lot of starch in it, but because the starch is composed of amylose, or a good chunk of amylose, and these alpha linkages are in amylose, we have a much easier time digesting starches than we do cellulose. So it's a subtle difference, but a really, really important one. Additionally, we have variations of these. So here's an example where we've got a mylopectin. And if we look at this main chunk right here, this looks like amylose, right? The one we just saw above. But what we have going on is we've got the side branching. So we've got additional branching going on. And in amylopectin, this occurs I think it's about every 25 glucose monomers, you get some sort of branch coming off the side. And this allows these molecules to essentially pack in a different manner. But really, this is just another starch derivative. So it's one of the many components that makes up starch. If you've taken human biology, you may have even talked about glycogen. We do the same thing in our liver. Glycogen looks a heck of a lot like amylopectin, but it has a lot more branching. It's almost every 6 glucose monomers that has a branch rather than every 24. So really similar with plants and animals, we try to store our energy in the form of carbohydrates that we can slowly release back during times where we need that energy back into our bodies or into our plant structure. All right, there's one last one I did want to cover, and this is chitin. This is a really interesting polysaccharide. If we look at it, it has all of these linkages, and all of these are beta linked. So again, our bodies aren't very good at digesting these, but there are some enzymes available where we can digest these, no problem. The main difference between these, though, is what's highlighted in green. These all have N-acyl groups coming off of them. So if we think about what that means, an acyl group is this chunk right here, and then the N just refers to it being attached to a nitrogen versus an oxygen, which would be an O-acyl. So in this case, chitin has a bunch of acyl groups and nitrogens off of it. These are found primarily in the exoskeleton of insects, a variety of crustaceans, and even in the plant walls of some fungi. So if you go on and take biology, you may talk about chitin. Plants and animals make a huge amount of this. It's one of the most common polysaccharides after the starches and after cellulose itself. All right, so that's pretty much it. Like I said, this is really just to prepare you for biochemistry. The main thing I want you to know from this section is the difference between alpha and beta linked, and to really remember that a glycoside or a glycosidic bond can occur between not only regular alcohols, but two monosaccharides that attach together and form a disaccharide or a polysaccharide. All right, so let's take a look at the end of the chapter. We've got a reaction summary here. I do want you to know the mechanism for ring closing reactions, right? So make sure that you know how to take an aldehyde, have it react with an alcohol to form a hemiacetal. I want you to know how to do this under a variety of circumstances. With monosaccharides, we saw a variety of reactions. Let me erase this really quick. The first one's really straightforward, right? We can acylate any one of those alcohols by using acetic anhydride. We can methylate or alkylate any one of those alcohols using methyl iodide. And then down here, we said we can make glycosides by reacting amines or alcohols with our carbohydrates. We saw over here we can reduce aldehydes with sodium borohydride. We saw that we can oxidize aldehydes with bromine and water under a specific pH. And then if we want to oxidize both our primary alcohol and our aldehyde, then we've got to amp up our oxidant to nitric acid, water, and heat. And then in yesterday's video, what we saw were the chain lengthening and chain shortening reactions. To be honest, I don't really expect you to remember the mechanisms for any of these, but I do want you to know the mechanisms that were covered in the video for the other ones. These ones aren't very synthetically useful. Like I said in the video, most of the time we just buy those carbohydrates commercially. All right, there's not going to be any skill builders due, so just make sure that you start studying for the ACS, and if you have any questions, feel free to email me.